Welcome back to the deep dive. Today we are tackling a topic that it sits right at the intersection of biology, survival, and I guess identity.
Hmm.
It's something that I think a lot of people have heard of, maybe even joked about to sort of cope with the frustration of it all.
Oh, for sure.
But they probably don't realize just how biologically real and measurable it actually is. We're talking about chemo brain.
It's a term that gets thrown around constantly in cancer survivorship circles. Chemo fog, brain fog, losing my edge.
Right.
And for a long time, and I mean, frankly, still to this day, in a lot of clinics, it gets dismissed. You know, people are told it's just fatigue or it's the trauma of the diagnosis.
The stress of it all.
Exactly, the stress of trying to get your life back on track.
The whole it's all in your head explanation, but, in the psychological sense.
Mm.
But what we're looking at today suggests that phrase is, ironically, kind of accurate.
How so?
It is in your head, but not because you're just stressed. It's because there is what looks like a visible biological scar on your DNA.
That's it. We're moving away from this idea of symptoms, and we're looking at mechanisms. And the research we have today is, well, it's honestly groundbreaking.
What's in the stack?
We're looking at a major longitudinal study. It's in Frontiers in Aging by Sayer and colleagues. And then there's a really compelling report from SciTech Daily about research from the University of Rochester and St. Jude.
And the headline here, it's pretty intense. The research suggests that cancer treatment might be literally fast-forwarding your biological clock. It's aging you at a cellular level.
It is a sobering concept.
Sure.
And we're focusing specifically on what's called cancer-related cognitive impairment, or CRCI.
CRCI, okay.
But we're looking at it through a very specific lens today. The adolescent and young adult patients.
Mm-hmm.
The AYA population.
So these are people aged, 15 to 39?
That's right.
They're not kids, not in pediatric wards, but they aren't geriatric patients either. They're in the absolute prime of their lives.
Exactly. So the mission for this deep dive is to unpack this hidden mechanism behind why their brains seem to slow down after treatment. We're going to look at this idea of epigenetic age acceleration.
Epigenetic age acceleration.
Okay, this is where we have to fundamentally change how we think about age. We need to move away from the calendar and look at the cells. We need to understand how treatment makes a patient biologically older than their birthday says they are.
And how that aging process actually messes with the brain.
Yes, and crucially, how it shuts down specific pathways in the brain, the neurotrophin pathways that we need for memory, for attention, for everything.
Okay, so let's unpack this. We start with this idea of the clock versus the calendar. I think for most of us, age is just math. Current year minus birth year. That's it.
And that's your chronological age. You're right, that's fixed. You can't change your birthday. The Earth has gone around the sun a certain number of times since you were born. But biological age, that's different. Biological age is a measure of the wear and tear on your cells. It's how old your body acts, or maybe more accurately, how much damage history your body has accumulated.
So you could have two people who are both, say, 30 years old chronologically. One might have the body of a 25-year-old biologically because they've been lucky and healthy, and the other might have the biological age of a 50-year-old.
Precisely. Think of it like a car.
Yeah.
You can have two cars from the same model year. One's been kept in a garage, driven gently. The other's been driven off-road through salt and snow.
They're the same age on paper.
But the condition of the engine, the chassis, completely different.
And in this analogy, cancer treatment, the chemo, the radiation, that's the off-roading through the salt and mud.
It is extreme wear and tear. And the Sayer study from Frontiers in Aging uses this concept of epigenetic age acceleration to basically measure that rust on the car.
Okay, epigenetic age acceleration. It sounds like something out of a sci-fi movie. We need to break that down. What is actually happening at the cellular level?
So to get this, you have to understand DNA methylation, the word DNM.
DNM, got it.
Think of your DNA as this huge instruction manual for your body. It contains every recipe for every cell you have. That manual doesn't change. The pages are printed in ink. That's your genetics.
Okay, so the text of the book is fixed.
Right. But your cells don't read every page all the time.
Mm-hmm.
A skin cell reads the skin chapter. A neuron reads the brain chapter. Epigenetics is the system of bookmarks, sticky notes, highlighter marks that tell the cell what to read and what to ignore.
Okay, that makes sense.
And methylation is specifically like a do not read sticker, or maybe a dimmer switch is a better way to think about it.
I like the dimmer switch analogy. So the DNA sequence is the light bulb, it's the hardware, but methylation determines how bright that light shines.
Exactly. And here's where it gets fascinating. Environmental factors, stress, Diet, and in this case, toxic chemotherapy drugs, can mess with those switches.
They can turn the dimmer down on genes that are supposed to be bright.
Or turn it up on genes that should be quiet. When we talk about epigenetic aging, we're measuring the buildup of all these chaotic adjustments, like someone went through the instruction manual and just started randomly gluing pages together.
And in this study, they use specific tools to measure that chaos, right? They mentioned things called GrimAge and PhenoAge.
Yes, and the names are pretty descriptive and a little intimidating.
GrimAge does not sound friendly.
It's not meant to be. It was developed by analyzing thousands of DNA samples to predict mortality risk. It's literally a death clock predictor based on these methylation patterns.
Whoa. So if your GrimAge is higher than your actual, chronological age...
Your risk of morbidity and mortality is statistically higher, yes.
That's heavy. And PhenoAge?
PhenoAge is similar. It predicts your phenotypic age based on clinical biomarkers. Again, looking at that methylation pattern. They're very sophisticated algorithms.
So let's bring this back to our young cancer patients, these AYA patients in their 20s and 30s. What happens when you apply these death clocks to them?
This is the core finding of the Sayer study. They looked at 51 newly diagnosed AYA patients, people with breast cancer, lymphoma, and compared them to healthy age-matched controls.
Okay.
And the results were stark. The cancer patients showed significant age acceleration. Their cells looked years older than the cells of their healthy peers.
And this wasn't just while they were, hooked up to the IV drip, right? Because I assume if you pump poison into someone, their body's going to freak out.
Right, that's a great point.
But does it stay that way?
That's the critical part. They looked at different phases: active therapy, recent therapy, and stable, which was 6 months or more post-treatment.
6 months, okay.
They found that GrimAge specifically was elevated during active therapy, which you'd expect. But here's the kicker. It remained elevated in the stable measures.
So even half a year or longer after the chemo stops, the biological clock is still running fast.
Correct. The rust doesn't just fall off the moment the treatment ends. The biological aging persists. Those dimmer switches don't just snap back to their factory settings.
That is terrifying. You survive the cancer, you ring the bell, you go home, but your cells are still acting like they're under siege.
It's a difficult reality. And this leads us to the big so what question. Why does having older cells make you forget where you put your keys? How do we connect a methylation tag on DNA to the experience of chemo brain?
Right. One is microscopic, the other is your daily life. What's the link?
This is where the Sayer study makes a really, really important connection. They didn't just look at the clocks, they looked at the fuel. They measured a specific molecule called BDNF.
BDNF, brain-derived neurotrophic factor. I've heard this called miracle grow for the brain.
That is the perfect analogy. Neuroscientists use it all the time. BDNF is essentially fertilizer for your neurons. It supports neuroplasticity, your brain's ability to learn and adapt.
It keeps things flexible.
Yes, and it helps existing neurons survive and encourages new connections. If your brain is a garden, BDNF is the water and the nutrient mix that keeps it lush.
So let me guess. As the patients got older biologically, the fertilizer just ran out.
That is exactly what the data showed. It was a significant negative correlation. As GrimAge, that biological age, went up, circulating BDNF levels went down.
Wow. It's like the accelerated aging is physically shutting off the supply line to the brain.
It is. But the study went even deeper. They wanted to find the exact mechanism. They identified a specific biological pathway. It has a technical name, KGG pathway, HSA.04722.
HSA.04722.
Catchy! I know, rolls right off the tongue. But it's also known as the neurotrophin signaling pathway.
Okay.
Think of it as a specific circuit board in the brain's communication network. This pathway is a chain of signals that tell neurons to survive and grow.
So it's the wiring that carries the grow signal.
Exactly. And the researchers found that the genes controlling this pathway, specifically genes like PIK3CD, HRAS, and RAP1A, were getting methylated.
Someone was dimming the lights on those specific genes.
Precisely. The epigenetic changes were effectively dampening the signal. These genes, HRAs, RAP1A, they're involved in signaling cascades, things like the MAPKIR pathway, which are essential for protecting neurons.
So it's a double whammy.
It's a double whammy. First, your overall level of BDNF, the fertilizer, is dropping because of the aging. And second, the cellular machinery that receives and uses that fertilizer,
It's being disrupted.
it's being disrupted by these epigenetic scars.
That is so thorough. You have less fuel, and the engine that uses the fuel is broken. It really explains why this cognitive impairment feels so profound. It's not just, I'm tired.
No, it's my neural circuitry is being starved and silenced. And that brings us to the real-world impact. Because if you're a 25-year-old survivor, this isn't abstract biology. This is your life.
Right. Let's talk about the symptoms. The Sayer study listed the specific cognitive domains that took a hit. This isn't just forgetting a name at a party.
No, not at all. We're talking about measurable deficits in response speed, executive function, attention, and multitasking.
Multitasking is a big one, and memory.
Memory was the most significant. In fact, they used something called the Reliable Change Index, the RCI, to measure objective changes. They found that the memory RCI showed the most significant number of these differentially methylated positions.
Meaning the genetic scarring was most obvious in the genes related to memory.
That's right. And this connects perfectly to our second source, the report from SciTech Daily. They really highlight why this is so uniquely devastating for this specific age group.
The adolescents and young adults.
Yes. This is the perfect storm concept. And you have to understand the sociology here. If you're 70 and you have some cognitive decline, it's difficult. But you're probably retired. Your life's pace has slowed down.
You aren't trying to launch a career or finish a degree.
Exactly. But if you're 22, you're trying to finish university. If you're 28, you're trying to establish a career, maybe buy a house, start a family. You need your executive function to be firing at 100%.
And suddenly your brain is aging decades in a few months. You have the biological brain of a 50 or 60-year-old, but the societal demands of a 20-year-old.
Exactly.
Yeah.
And the Williams study points out that these cognitive deficits, combined with the immense pressure of early adulthood, lead to significantly worse educational and employment outcomes.
Compared to their peers or even their own siblings?
Yes, they're trying to launch their lives with an anchor tied to their ankles. It can create a socioeconomic gap that lasts a lifetime.
The Williams study also made an interesting distinction about the type of treatment. We often just say cancer treatment, but they found a difference between chemo and radiation.
They did, and this is important for patients to know. They found that chemotherapy was linked to the most rapid biological aging.
Really? More than radiation. I always think of radiation as this, high-intensity destructive beam.
Radiation is intense, but it's usually targeted. Unless you're radiating the brain directly, the damage tends to be more localized. Chemotherapy is systemic.
It goes everywhere.
It travels through the blood, goes everywhere. Every cell gets a bath in that toxicity. It causes widespread DNA damage, these things called double-strand breaks in the DNA helix.
Wow.
And this forces the body to go into overdrive to repair it all. That repair process is exhausting for the cell. It just accelerates that rusting process we talked about across all your tissues. It's a systemic fast-forward button on the aging clock.
That is so heavy, and I can feel our listeners tightening up right now. We've established that the clock speeds up, the brain fertilizer runs dry. It sounds like a trap.
It does.
But before we leave everyone feeling hopeless, we have to pivot. We've unpacked the problem. Is it permanent?
This is the most important part of the conversation. If you take nothing else away from this deep dive, take this. Biological aging is not the same as chronological aging.
You can't turn back the calendar.
You cannot be 22 again if you're 32. But biological aging is malleable. It's plastic. Because it's based on chemical tags on the DNA, that methylation, it can, in theory, be modified.
So, we can sand off the rust.
To an extent, yes. The researchers at Wilmot Cancer Institute and St. Jude are looking at exactly this. They suggest that lifestyle interventions can help slow or even reverse this clock.
What kind of things are we talking about?
Well, it sounds basic, but things like cessation of smoking are huge. But the big one they cite, and the one I get excited about, is physical activity.
Exercise. It always comes back to exercise, doesn't it? I feel like every time we look at health data, the answer is move your body.
It really does. But here, we have a mechanism for why it works. Exercise has been shown to have value in reversing epigenetic aging. Why? Because it increases BDNF levels naturally.
So you're manually replenishing the fertilizer.
Exactly. When you exercise, especially aerobic exercise, you are signaling your body to produce more BDNF. You're helping to clear out cellular stress. You're turning the dimmer switches back up on those neurotrophin pathways.
So it's not just feeling better, it's actually changing the readout on that GrimAge clock.
We're talking about changing the readout.
That's right.
And this is incredibly empowering. It moves the narrative from, I have permanent damage, to, I have a manageable condition that I can influence.
It gives agency back to the patient.
It does. And nutritional improvements play a role too. Anti-inflammatory diets can help reduce that systemic stress.
What about on the medical side? Are we going to see this in clinics?
I think so. The research is shifting toward timing. The Williams team is investigating the ideal time to intervene. Is it during treatment?
Mm-hmm.
Immediately after. The goal is to catch this acceleration before it becomes a permanent trajectory.
Right, instead of waiting for the patient to come in complaining about memory loss 2 years down the line.
Exactly. I think that's where we're heading. Imagine a blood test for brain health as part of your standard survivorship care.
Walk me through that. What would that look like?
Well, instead of just checking for cancer recurrence, your doctor draws blood and checks your DNA methylation and your BDNF levels.
Huh.
And they could say, hey, we see your CNS is spiking and your neurotrophins are dropping. You are at high risk for chemo brain. We need to get you into a cognitive rehab and exercise program right now.
To protect your brain before the fog even rolls in?
That would be the game changer. It validates the patient's experience with hard data, and it offers a proactive treatment plan. It treats the brain as a vital